It's really great pleasure to be back here again. I think we were here in 2020, actually giving a demonstration on the type 2 diabetes knowledge portal. So I'm very happy that you invited us back. And I love being part of the dkNET network. So it's my pleasure to introduce today's session, and then I'm going to hand it over to MacKenzie. He's going to do the lion's share of the demonstration today. I'm thinking folks can see my screen, and I'm going to switch to slides. So that's great. Beautiful. All right. So I wanted to begin with a little bit of a motivation and I'm going to walk you through the background before MacKenzie takes you through the demonstration. And as Koei said, we'll have a nice part of the session for Q&A. But if you have any questions as you go, you can pop them in the chat. And I might answer them as MacKenzie's going, so we can make sure it's live and dynamic, so that you feel like if there's anything you see along the way, feel free to flag us so that we can make sure we're being clear about things. So I thought I'd start with why we're here and the foundational program that funds this activity, and it's the Accelerated Medicines Partnership for Common Metabolic Disease. And many of you may have heard of the type 2 diabetes AMP program, which was the beginning of this, and we expanded to common metabolic disease in the last 2 years. This is a public-private partnership between the NIDDK and the FNIH, and for industry partners, Amgen, Lilly, Novo Nordisk, and Pfizer. And our goal is to advance the use of human genetics in study into defining new medicines and therapeutics. And we do this across 6 large-scale disease areas across common metabolic disease. And really, the foundation of this effort is in human genetics. And I say that because, that's where we believe that there's a strong, foundation for drug discovery. There's a traditional approach for drug discovery, which is target identification through biomarkers or cellular and animal models, which then brings you to relevance in human. But you can reverse that and discover these loci and biomarkers in humans, explore the mechanism, and then bring it back to humans for study. And there's a lot of efficacy and study in clinical models that have human genetics at its basis, and in terms of all the different areas for modulation, relevance, causality in humans, and mechanism action. Human genetics is a very effective approach, right? So our goal is to use this as our backdrop as we try and find better targets for these common metabolic diseases. And this started, way back in the early GWAS era, in about 2015, when we developed the type 2 diabetes knowledge portal, which had a simple mission, this was in the time of large-scale GWAS, genome-wide association studies, or sequencing studies where people studying very large cohorts and producing, summary statistics of association for diabetes and related traits. We wanted a way to make that accessible to someone who's not a geneticist. So the goal with this portal was to make the genetic data accessible. And since then, you might be feeling, if you are a basic researcher or someone in an industry setting, that it's sort of the genetics world is sort of a tsunami of associations for GWAS loci across complex disease, right? Where there's regions of the genome all over the place. In fact, there's hundreds of thousands of loci associated with complex diseases. And this just gives you a sense of, for some very big trait areas, how many loci have been associated. I think this number is even going up. I'm a little out of date here. But if you were to study these, it would take a lifetime, right? And our goal is to try and find better medicines for people with these chronic diseases. But the good news is, I believe our field is moving in the right direction in terms of generating the right data, creating novel approaches to mine these data and make them accessible to others, and to generate the relevant tissues and cell types and perturbational experiments to understand the mechanism of action. But you also have these wonderful foundational resources that are repositories for the relevant data, like the GWAS catalog, which does what we did with the type 2 diabetes knowledge portal way better. They do this across all common diseases, and it's a repository where you can access all the summary statistics. And there's other resources like that. Moreover, our foundational resources for technological and computational advancements in genomics, right, for you to understand basically the gene function and different things about the gene, and DNA and epigenomic annotations. These resources are also far more advanced, right? And you have visualizations and representations and repositories for all these, with like Roadmap, GTEx portal, ENCODE, and more recently, the single cell. So these are resources that span diseases and tissues, but make repositories for relevant epigenomic and perturbational and model system-based studies. However, how do we go from here, which is a locus associated with obesity in the specific region around FTO, to a single variant with a mechanistic hypothesis? This is the challenge, but we have to do this for hundreds of thousands of loci. But we're basing this on a simple model that genes predict organismal function, right? But if genes are regulated by regulatory elements or cell states, but variation in our DNA governs those. And so our goal is if you can, figure out what all these causal variants do, you might be able to elucidate an organismal function. So inherent in that are many questions, right? What is the variant at a given GWAS locus? What is the regulatory effect? What tissue is it active in? Importantly, what is the gene? What is the gene in this locus that I should be studying? If you can also get lucky, you also learn about the different pathways that may be involved. And finally, what is the mechanism of action, which helps get you to a target or a therapeutic? So our goal is to extend our current platform to facilitate this effort and catalog and aggregate all the necessary data and resources, make them accessible and visualize them. And the goal in here, not just to be repositories because other places do this better, but to create a way to represent integrated results based on scientific questions. And that's really the key of the Knowledge Portal and what you're going to hear from MacKenzie today as she goes through it. So this is the portal. She's going to walk you through it shortly. Been around since 2015, as I mentioned, and in 2020 is when we expanded to the Common Metabolic Disease Knowledge Portal. And this just gives you a sense of the growth over time, where it started as a very simple resource, but now has really become sort of a sustained and sort of resource that has more and more questions you can ask scientifically about genetic and genomic data. So what's nice is now you can begin to address different questions in the form of workflows. Things like what variants contribute to a signal at a given locus. For a given region, what variant or variants might I study or put forth for functional characterization? So MacKenzie's going to walk you through how you might do that. What pathways are enriched for my favorite gene or phenotype? And what are the likely effector transcripts for a host of cardiometabolic phenotypes, type 1 diabetes, type 2 diabetes, and so on and so forth. So that's what we're going to try and address today as you walk through the demonstration. But first, a little bit about the foundational data. So there's three major core data sets that sort of power the resource. And the genetic data is representative of over 50 countries. And what's nice about that is while we're still, overrepresented with Europeans, we're slowly getting better at representing other populations, because I think that's incredibly important in terms of how we understand human genetics across the earth. And the other foundational data set are genomic annotations. So this helps you then, determine what the cell state or tissue is. And this, we partner with the Common Metabolic Disease Genome Atlas to bring genomic annotations that are warehoused there to the portal and integrate them with the genetic data. Currently, there are 77,000 genomic annotations at CMDGA, but we represent about 5,500 of those. And these are specifically annotations relative to genes linked to cis-regulatory elements and cis-regulatory elements themselves. This helps you understand, the regulatory effect of a given variant, because most of these are in the non-coding region of the genome. So this is another foundational data set. However, how do you use these data sets, right? Well, before one thing to mention, these annotations are enriched for cardiometabolic cell types and tissues. So one thing that's nice, the pan disease resources like ENCODE focus on all tissues. Our resources really tends to try and get tissues and cell types that are specific to cardiometabolic disease. So you can see we're slightly enriched currently for that, but we hope to increase that over time, getting the relevant tissues and cell types for common metabolic disease. Then to pull this all together, you have the genetic data and you have the functional genomic annotations. But to make sense of it all, if it's just sitting independently, you really want to run bioinformatic methods or computational approaches across both these data sets to produce mechanistic hypotheses and results that you can actually use and make the results, visualizable and accessible. And this is a host of methods. MacKenzie's going to talk about them a little more, but ideally, this is what makes the results useful within the Knowledge Portal. And so again, these questions will be tackled today as we go through, and then I'll return to this at the end, but I want to turn it over to MacKenzie right now, who's going to walk you through the actual resource and sort of take you through each of the different workflows that address some of the scientific questions with the data that I spoke about. So with that, MacKenzie, I'll turn it over to you.
All right. I'm sharing my screen. Can everyone see this all right? Okay, great. So as Noël said, I'll be walking you through the CMDKP today. So to identify new targets and to improve the accessibility of the data, the Common Metabolic Disease Knowledge Portal, or CMDKP, was developed. The CMDKP is an open access resource that presents integrated and analyzed genetic and genomic data to spark insights into common metabolic diseases and traits. Today, I'm going to walk through how to use the resource, along with how users can ask specific scientific questions about genetics and genomics, and to also view integrated results and visualizations to address questions or shape hypotheses. So starting out here on the home page, in the top right, we have this navigation bar. If you select on a certain tab, like for example, Help here, it actually brings you to the Knowledge Portal guide, which gives information on the tools, visualizations, and various workflows in the knowledge portals. On the right here, we have a table of contents of other topics that you may be interested. Selecting these will hyperlink out. Underneath the search bar at the bottom, you can learn more information about the portal itself. You can read our latest news items. You can view a step-by-step guide or watch a tutorial. You can sign up for our quarterly newsletters and other updates, and you can also learn more about how the project was actually developed. One of the 4 types of data that we represent, the most important are the genetic and genomic data. To access the full summary of data sets, under the Data tab in the toolbar, you can select Genetic Association datasets. But before I bring us to this page, I just want to take a moment to actually talk about the data that live within the CMDKP. The CMDKP represents 422 traits. Our genetic data includes full GWAS summary statistics from 389 datasets. More than half of these were obtained in collaboration with disease-focused researchers, and others are from the GWAS catalog and other public sources. These data will only increase and mature with our collaboration with the GWAS catalog. In order to determine which variants to prioritize or genes to study from a GWAS locus, integration with tissue-specific annotations is a critical data component. Our sister resource, the Common Metabolic Disease Genome Atlas at UCSD, is built as a repository to warehouse raw data from epigenomic annotations, which are obtained from individual investigators, or they're curated through large-scale resources such as GTEx and Roadmap, as Noël mentioned earlier. We can integrate these results directly into the CMDKP. To date, we have tissue-specific community-derived epigenomic annotations from 5,500 datasets. Now, another set of components is curated lists of data. From GWAS studies, we have curated credible sets, which are often not accessible. Getting these from the community is not a trivial task, and it's important to have these at scale. Our goal is to get curated credible sets for every disease area, and we've worked with experts to obtain curated credible sets for 45 traits. In many cases, each disease community could also create a customized effector gene list. These are also not readily available. We are able to integrate these dynamic genetic and genomic lists directly within our resource. Currently, we have 19 represented on our site. So as we go back to the home page, I want to bring us back to the Data tab here, where you can select the genetic association sets. By selecting on this, it brings you to the Genetic Association Dataset page, which includes 400 traits. The dataset page first brings the new datasets ingested as part of our new October production version, with older datasets below the page here. If you're interested, you can click on a specific dataset, which is hyperlinked in blue on this side here, which gives information such as contributing cohorts and other publication information. We also give you the option to download summary statistics and view the specific paper that dataset was published in. Other features on here, you can see whether the dataset is open access or pre-publication, how many samples are in the dataset, the specific ancestry, the technology that was used, and if there was a specific contributing community, which you can see with the icons down here. This page also has various filters, and one that I'm showing here is the ability to filter by phenotype group. As I walk through the CMDKP, I'll show you how these data are specifically integrated. So now we have our epigenomic data, and to access these, you can click on the same Data tab here and select Genomic Data Sets. This page is the summary of the annotations that we represent from our sister resource, the CMDGA. These are organized by disease group. The accession number, which can be seen here, actually links out directly to the CMDGA resource itself. So I'm going to spend a little bit of time talking about the CMDGA, which is an open access functional genomic portal that provides the collected and processed sequence data from thousands of functional genomic assays. This is what the homepage looks like. The CMDGA houses around 7,000 total genomic annotations. The CMDGA represents cis-regulatory elements, variant effects on CRE activity, genes linked to CREs, variant effects on gene activity, gene expression, and function. Of the genomic annotations, about 5,500 are integrated within the CMDKP as of October. Of the 5 classes of data that I just walked through, we represent cis-regulatory elements and genes linked to cis-regulatory elements. So I want to show you this slide here where in graph A, we see the tissues and annotation types distilled across the portal. On the bottom of the graph here, you could see the tissues and various types of annotations are represented by the different shades of red here. The portal integrates chromatin state and candidate regulatory elements more than any other type. In the graph B over here on the right, you can view the cell and tissue types in the CMDKP versus ENCODE. The graph shows that we have a higher enrichment in tissue specific for CMD than ENCODE as a resource is pan-tissue. Again, I know Noël mentioned this earlier, but I'd like you to take a look at the kidney, muscle, and cardiovascular here, which we have high enrichments for. So our resource is not a repository. We aim to make these data useful and integrated by running a suite of current best practice methods for genetic and genomic data. I've listed on this slide what we have implemented currently across 5 areas: genetic association analysis, prediction of variant effects, gene level association analysis, annotation global enrichments, and SNP to gene. Few resources have incorporated these many methods. So we can apply these methods at scale using the following pipeline. Starting with the data intake process, we work directly with collaborators, consortium members, and the public to aggregate all of this data. The process is the same for both genetic and genomic data. Our next step is the data goes into the aggregator where we store summary statistics and other data that we receive. This knowledge base is built off of Spark and Hadoop and runs bioinformatic methods. These results are then stored in the bioindex. From there, our visualizations are built to make the data accessible to the users. I will now walk you through how to view the various workflows and results within the CMDKP. Here again, back to the home page. You might be wondering, now that we have all of these data, how can we make them accessible to the user? Through the home page here, you can search the CMDKP first by gene, region, or variant, by your favorite phenotype, or you may be interested in looking at disease-specific portals. I'm going to start here by searching for the type 1 diabetes phenotype. This brings me to the type 1 diabetes phenotype page. The search returns genome-wide associations statistics for a novel sample overlap aware approach called the bottom line analysis, which is applied to all phenotypes. This type of bottom line association meta-analysis is not offered by any other resource. Our page includes features that commonly represent these data, such as the Manhattan plot here on the left and the QQ plot here on the right. Underneath the first visualizations, we present to you the top single variant association signals for type 1 diabetes in this card here. If you're interested, you can enter into the individual data sets that were used in the meta-analyzed results to generate the bottom line associations. Again, you can filter through this table and these link out to the individual data sets. Here in this card, we also show the top common variant gene level associations for type 1 diabetes. This information is calculated from the bottom line genetic associations using the MAGMA method. This table represents the top pathways for type 1 diabetes, which are calculated using the MAGMA algorithm. Next, you can view genetic correlations for type 1 diabetes. Genetic correlations are important because they help prioritize likely causal relationships. We have calculated these statistics based on recent open access methods. Last, on the phenotype page, we represent the globally enriched annotations for type 1 diabetes. These annotations come directly from our sister resource, the CMDGA. So I'm going to take a moment. Is there any questions on what I've presented so far? I know that's been a lot. Okay, I'm not seeing any questions, so I'll continue on. You can also search for a region from the homepage using the same search feature here. The region page shows all genetic associations across all traits in the portal, along with epigenomic annotations across that region. So if you search for a gene name like FTO, the region on here is defined as the coding sequence of the gene plus or minus 50 kb. So if you're interested, you can customize this region by expanding it to the distance you'd prefer using this drop-down menu at the top of the page. The items that appear on this card have text that are blue hyperlinks. This first link, which shows bottom line integrative analysis, gives more information into how the bottom line analysis is run. And for example, here, the second link shows a complete guide of the region page. I want to also bring your attention to the circles with blue eyes. This is a tooltip. If you hover over these tooltips specifically, you can see detailed information regarding the card. This tooltip, for example, gives information about the search results. These genes in this section here are located in or overlapping with the region that I searched. The plot below here shows the most significant genetic associations in this region. Each color on the plot represents a separate phenotype group. By clicking on View Associations by Phenotype Group here, changes the view to see a summary of bottom line meta and analyzed associations for phenotypes in this group. Again, as you can see, each color represents a different phenotype group. The table under the PheWAS plot presents the lead variant in the association based on LD clumping. This table provides the phenotype group, and other information related to this. By selecting the Show Variants button here, we provide information regarding the LD proxies. The next section on the region page is the Genomic Region Miner, which provides a region-based locus zoom view. You can customize this view using your favorite tracks. I'd like to note that the plot automatically selects the association for the top phenotype on this result, which can be changed as well. As you can see right now, it's set to body mass index. Our last section on the region page provides a table of all variants in the region. One of the major challenges are how to prioritize variants in a specific region. I'll explain how the CMDKP can help users do this using the variant Sifter a little bit later on in this presentation. Headed back to the home page, I am now going to search for a variant. From the region page, I searched for the lead variant for FTO in the search box, which you can see here. As a result, I am brought to the variant page. The variant page addresses variant impact predictions and genetic associations. The closest gene card here presents the nearest gene or genes to the variant that you search, predicted by the ensemble variant predictor. Beneath the closest gene card is the predicted variant consequences. The consequence shows the highest impact consequence of this variant on the indicated transcript. The most severe consequence is highlighted in green on this table, as you can see where I'm hovering here. If you're only interested in a certain impact, let's say for example high impact, you can select this button specifically to filter the results in the table below. The first visualization on the variant page is this PheWAS plot, which represents the lead variant and other variants across phenotype groups. Again, each color represents a different phenotype group, and each triangle here on the plot represents a specific phenotype. The triangle direction indicates direction of effect. If you notice, across the core pages of the portal that I've shown so far, it automatically shows the results for all ancestry. But if you were to select set page level parameters here, you can select an ancestry of interest. By clicking on it, you have this drop-down menu, which shows the available ancestries you could set this to. And by selecting Go, it can change the page to that specific ancestry. If I headed back to the table underneath the PheWAS plot, I want to show you if you're interested in seeing which individual data sets were used to calculate the bottom line p-value, you can navigate directly to them by clicking on the datasets button on the table. If you were to select the button for top 25 variants, you can actually see the top variants in this phenotype group. Okay, so I see there's some activity in the chat. So I'm just going to take a moment to see if there's any questions that I can handle right away.
Yeah, MacKenzie, I think Eve has a couple good questions related to the region page. And I'm looking for information for BMI and also about the ancestry. So Eve, do you want to unmute yourself and ask that? I think we have time for that.
Sure.
Okay, the ancestry I'm interested in is going to be North and South America, Middle America. And I didn't, it may have been on there and I just didn't see it because it's pretty small. What do we got? Let me get the chat out of my screen.
I just went back to that slide that I had this information of the ancestries that we have currently.
So I mean Hispanic. Okay, so I'm actually from New Mexico.
Mm-hmm.
And so a lot of people here can tell you that they're from Mexico or Central America.
Right.
Does that make sense? So
yes.
I don't know where they don't consider themselves Hispanic or Latin American.
Mm-hmm.
I just want to warn you. And that can be offensive. They consider themselves indigenous and Native Americans. So, okay, I'll mute. Thanks.
No, it's a very fair point. I think, I think these are the ones that we have represented currently in the portal. And then, we have the specific information in all the drop downs for the specific cohorts to contribute. And this is how we've had to group them based on their representation. But you're absolutely right. As we get those more represented, we will ensure to refine them and have them more specific and appropriate.
Mm-hmm.
It's a great point. Great.
Thank you for that question. I see some other ones, but I think we'll hold off until the end for that. Is that correct, Noel? For some of these. Oh, you're on mute.
Wait, what about BMI, Eve? Do you want to ask that one?
Sure.
Too?
Let's wait and do that at the end.
Okay, sure.
We'll get back in that screen. Okay.
Okay, great. Thanks, Eve. Okay. So I'm taking you back here to our homepage. And here you can search for a gene of interest, and I'm going to be searching for SLC30A8. So here is the SCL30A8 gene page. The gene page centers on the coding sequence of the gene and the biological role of its product. The first card at the top shows the functional associations for SLC30A8. The first plot on the gene page shows common variant gene level associations for SCL30A8. This plot can be filtered by ancestry, which can be seen here. So where we have sequence level data, we can present the rare gene level associations with masks that can be applied to the burden statistics. So if you were to select this mask plus plot button here, you can see that 7 different masks were applied to filter the variants used in the association analysis. In another spot where we have sequence data, we also can provide a huge score. This huge calculator module creates a posterior probability to support the involvement of SLC30A8 in a disease or a trait. This result here shows that there is compelling evidence for the involvement of SLC30A8 in type 2 diabetes, which is shown here. On the home page, you may also want to enter and explore disease-specific portals. You can enter into one of the 5 disease-specific portals from this tab here. Using the same software as our established Type 2 Diabetes Knowledge Portal, we have created community portals for cerebrovascular disease, cardiovascular disease, sleep disorder, and type 1 diabetes. Here is an example of what this community portal would look like. The community portals have the same features and tools as the CMDKP, but only present the user with disease-related traits and curated content by this specific community. The portal also has various tools to help users learn more about the data and explore hypotheses. By selecting KP Labs from the navigation bar here, you have access to cutting-edge analysis tools that are still under development. I would like to note that the data supporting these tools are accurate, but the tools themselves are in beta. These tools are available for early access as we hope to gain feedback and suggestions from the community for further development. So everything so far that I have shown has been focused specifically on how to visualize and explore results from the genomic and epigenomic data. We also have more sophisticated types of visualizations and tools that may serve a different audience, specifically those who may want to use a dense tool to prioritize variants, or potentially individuals that just want to know what genes the field believes contributes to human disease. So what if you're particularly interested in a region and want to prioritize variants for a specific functional study? We have a new tool called the Variant Sifter to help prioritize these variants. This can be accessed on the homepage on the KP Labs tab at the bottom here, or through the region page. I'm going to take a moment to describe how this specific paper attempted to answer a functional hypothesis for this specific variant and gene. This example is based off of the following paper on how they specifically applied the GWAS translational model. This paper combined different evidence sources and generated data to create a hypothesis for functional variants associated with LDL cholesterol. They specifically used GWAS data, ETL data, chromatin interaction data, annotations, and transcription factor binding sites. I will now show how the variant sifter integrates the same relevant data types to address the same hypothesis. So here is the homepage of the variant sifter. So the first thing I'm going to do is build my search criteria. Here I am searching for the LDL cholesterol phenotype, and in this box here, I'm searching for the RRBP1 gene. So by selecting search, I am provided with a result that has 11,000 variants. So with this number, how do you know which ones to prioritize? Well, first, with the GWAS data that's presented, you can filter according to variant ID or p-value, for example. And to help prioritize, we are going to add in additional data types through this tool. As I'll be moving further down, it'll help whittle our list to prioritize these variants. Adding in specific credible sets narrows the list as fine mapping indicates the most likely causal variants contributing to the signal at a locus. I've selected here the Graham 2021 lipids from this drop-down menu because the paper used this credible set in their hypothesis building. Here, you can select an annotation that's enriched for your tissue of interest using results generated from Gregor. I have added enhancer here based on the global enrichment plot. This enhancer, which you can see in the purple, is enriched in the liver cell. So I've selected this tissue underneath here on the track. This selection shows that through SNP to gene methods, you can see which variant is linked to a gene. By selecting liver LDL from the drop down here, we can actually see that the gene I searched for, RRBP1, is linked to our variant of interest. As you can see, this line here that marks the variant is intersecting with this track. The results table at the bottom of the page indicates here that my top variant is actually associated with my gene of interest. In this results table, you can also view specific evidence for credible sets, as you can see here, biosamples, genes, and more. But I want to take a moment to look at the credible sets evidence. If you look here, there is a hyperlink with the accession number to this data set. And by clicking on this hyperlink, you're taken to the summary for that specific annotation file. So as you can see, there's all this information regarding the data. And if you're interested, you could actually select the publication that these were published in. So while this result was expected and I saw what I wanted to in this results table, I would like to note that there are caveats. This resource needs the right evidence and sources, so we will need to continue to add more data. Non-expert users also need to prioritize variants. Biologists and members of the pharmaceutical industry may be interested in understanding what genes the community believes cause human disease. Effector transcript lists are a powerful resource to guide the non-geneticist community. These are complete with underlying data in the portal. So if we were to go back to the home page here, you can actually access these lists through the Tools tab on the navigation bar. We have identified over 50 papers that were able to do this. Here's an example using pre-publication results from the Cardiogram Plus 4CD Consortium. These results can be buried in supplemental tables in the form of static spreadsheets like these. And if you look at the bottom here, there's multiple tabs that have data and you can't link between them. These are remarkably important on top of all of the data that we've already loaded into the portal. So how do we make these useful? Using the same paper, here's an example of our interactive representation of the effector gene list. As you can see here at the top, our table provides an interactive graph, and the table below has links out to our data in the portal. So I've highlighted here the nearest genes. So if you were to select the ski gene, it would bring you to the actual page on the portal. We also incorporate the methods used to generate these lists. We have 19 contributed and curated effector gene lists across each biological system. We also have lists produced by bioinformatic prediction algorithms. Our goal is to represent all effector gene lists for all disease communities and those published in current literature into our resource. We also welcome collaborations with contributing data or methods, and you can contact us if you are interested. So I'm going to take a moment. I see there's some activity in the chat. If there's any questions on what I've presented so far, I can also go back in the slides if there's anything of interest. Okay. I don't see any chats or anything in the Q&A. Am I missing any?
No, I think, how is the effector gene list for type diabetes? Monica, great question. Monica wanted to know about our effector gene list for type diabetes. Actually, Monica, there's two right now, and they're both unpublished, but they're both pretty excellent in terms of what they do. Actually, MacKenzie, would you mind actually going to a live demo? I think we have some time for that. Why don't we show Monica where those are?
Okay, give me 1 second. I just need...
So one is produced by Kyle Galton's team, where he basically did a similar approach to one of the heuristics we have on the portal right now. He took regulatory, genetic, and perturbational evidence, weighed it created a heuristic, and we represent that on the portal with all the supporting evidence. And same with Steve Rich and colleagues did the same thing. And we actually have that also represented from their work. And what we're hoping to do actually for the next year is to overlap these two and see the similarities. It turns out they're not completely overlapping, which is interesting because I think it's about how they weigh different evidence. Here's Kyle's list. So Monica, this is the one that Kyle generated with his relevant evidence, and he has like a rating scheme as well. This is something we're hoping to write up as well with Kyle. We're working on it to write up a manuscript about the entire type 1 diabetes knowledge portal. And so you can see the top gene. Again, this links out to the gene page, to the locus page. And then over here on the view, you get the full specifics on the underlying data, and it will bring you either to OMIM or MGD or any of the relevant model system databases, or to CMDG itself, where you can find the QTLs or epigenomic annotations themselves if you want to get to the underlying data. So that's where that stands. Let's see, anything else on that? Here we go. So Eve is asking novel gain of function GC. Oh, sorry, I just lost went past it. Sorry, let me get back to it.
Also,
Novel
can everyone see my
function.
screen?
Yeah, we can see your screen. Yeah.
Okay, perfect.
If you were looking for something to demonstrate, so Eve, you want us to demonstrate that. So let's go to GCK, or there's a variant. I'm not sure we can find it by that. We may be able to have to look at the RS number, but we can look up that to demonstrate. I'll pull the paper. Great. Is there a graphic to contrast? Great question, Monica. We actually want to make one because we have a nice graphic to contrast the type 2 diabetes ones we have, but we need to make that for this. So that'll be in the next release. Good question.
Okay.
So, I saw Eve have several questions.
Yeah.
You're going to answer the last one, right?
I got to get to that gene. So GCK, let's see, let's go to, let's see if we maybe go to the gain, it's a gain of function. So I guess if you go to the gene page,
Okay.
if I find it, scroll down, you'll look for the functional consequence of the variants. Maybe it's in rare variants. Let's see what we got.
Still loading. We have a lot
Still
of
loading.
data.
Yeah, so for those who were watching the recording, I think Eve is asking to do a demonstration using a novel gain of function GCK variant P variant 4552, C455L, and it's associated with type 2 diabetes. Yeah. So
MacKenzie, I'm having trouble looking screen. I probably should have been doing this.
Oh, it's okay. Do you want me to stop sharing and you can share your screen?
Yeah, because I keep on thinking it's my screen.
Okay, sorry.
No, it's my fault. Let me see what I can do.
Okay, there you go.
This may take a little bit more time, but what's your question about it specifically, Eve, while we're getting to it?
yeah, if you can unmute yourself if you would like.
So I see in the chat, she says, it's just one question to demonstrate they believe it confers the owner with the larger cells.
Yeah, let's see what it is. Let me get the paper back. Here we go. Here we go. Let's see. This is
TTD. Oh, wrong one. I didn't want to go to that. I wanted to look at the variants. It's just loading a second. Do you have an RS number I can type in? That would be way faster.
No. Okay, let me get this for you. Okay. Oh, for God's sakes.
Because it's a functional variant, but
Well, you know,
so it's associated obviously with many different phenotypes. I'm gonna look at other questions if you have them. Here we go.
yes, it will be.
Oh, this is you in the chat. But what did you want us to demonstrate with it specifically?
Let me see if I can unmute. So can you find it? Did you found it? Oh my goodness, you did find it.
Well,
Thank
I
you so
found the
much. I'm
gene,
gonna mute. I
not
just
the
thought
variant.
we would, hang on, let me mute because I have a really crummy connection at home too. So
Oh, okay.
yeah, I put it in the chat, the paper it's from, just so you
Okay.
have it.
Great, thank you. So this is obviously, the gene itself is very strong. We can figure out we can view evidence review calculator because this may be useful here. Let's see if I can figure out it's loading for us. Any other questions while things are loading? Let's see.
Yeah, there is a question, actually it's previous submit.
Yeah.
So, let me check.
It's
Is it possible to upload a list of genes to get association for them from your portal?
yes and no. So there's two ways to do it. The first way you do it is there's one sort of little clunky way, but if you go to your favorite region or gene, you see this little button. You could download, you could go here to this, you could download, if you're at the region you want to download right here, what will happen is if you hit this button, you will download all the relevant annotations and associations for this region plus the associated phenotypes. So you can get all those results. Or you can go directly to Linares. It's an app where basically you can put in a set of commands that are pretty easy with lots of instructions for it, where you can basically get for whatever region you want or set of genes, the associations, all the annotations, basically any result that we have indexed in our bioindex. Also, Bioindex is also available for you to query. It's a little new in terms of its usability, but it is actually our entire bioindex can be queried on the command line. This gives you a little bit more user-friendly way of doing it, which we built out. But now that we have Bioindex sort of fully open access, we're going to build up all the documentation for that. So you can do it through the command line currently, but not necessarily right on the site.
Thank you. And there is a second question. So is the data for genetic association is available for HG19 genome build?
Not right now. We are on, what build are we on? We are on HG. Yes, sorry. Oh, you're all HG19. Sorry. Yes, I was thinking, yes, we're all HG19.
And she said, I'm looking for genetic association for T1D that were mapped to, I think, the cronk 38. So she's looking for the data for genomic association with T1 type 1 diabetes available for HG19. And then she said she's looking for genetic association for T1D that will map to, I think she means the 30, cronk 38. Yeah, let me see if she's here.
The new builds, so we're only on HG19 right now. Is that the question?
Yeah.
So this is the type 1 diabetes knowledge portal, which has all the associations for type 1 diabetes and its associated traits. It's just a different lens as MacKenzie described, but it's the same platform. It's just if you want to view the content specific to type 1 diabetes, it's there. And then you can pop right back out to the CMDKP. Any other questions? One thing I want to mention is, well, two things I want to mention. One is if you're interested in any more questions, I wanted to make sure that you can get a hold of us very easily. That's important to us. You can email me or MacKenzie directly, right there is an email, or you just go to the contact form up there, help@kp4cd.org, or you can attach yourself to our mailing list if you'd like to get mailing list updates. We don't spam you a lot. We get back to you within 24 hours. So we'd love to hear from you. This is the team that builds this wonderful resource across many sites and countries. It's a pleasure to work with them all. And then one thing I thought I'd mention, which I think is very useful given sort of what's going to happen with all of our data and results come 2023 based on our NIH requirements to make data even more accessible than I think a lot of us are doing already, is part of what we've done with the KPN and building all these portals is people have his specific types of data that doesn't completely fit within our current visualizations and things, but we really enjoyed working with people. So we built this thing called bring your own results. The idea being that you can bring your own results, anything genomic results, that is. And we have a suite of classic visualizations that you can customize and build for your own portal. And what's nice about that is once you did that, you can make it accessible for publication, for reviewers to look at, You can integrate it into our portal. You can also, and this is my favorite piece, is let's say you have genetic associations for your favorite phenotype that you generated, but you want to see them with annotations that we have from the CMDGA. Well, within the resource, you can actually customize that and build a portal associated with that. And then you can make it public, or you can integrate it with our current portals. This is something we built as part of being able to try and work with researchers to get results out faster, and also enable, a lot of 1,000 flowers to bloom, if you will. This is to help propagate a lot of portals in this area, not only for complex disease, but also for specific areas. Like we have a whole portal devoted to the APOL1 gene. We have a portal devoted to VQTL results. We have a portal devoted to genetic clustering for type 2 diabetes. So with all these heat maps and things like that. So it's the idea of making fast access to results easier without building a heavyweight portal that takes months. You can build these in days, and you don't have to be a programmer. So that's one thing I thought maybe you might be interested in. These are the portals we built. You can take a look at them. All of the effector index pages are built in that framework, just to give you a sense. So that's something you may be interested in if you have results, and we'd love to collaborate you. If you have like something special that you're like, I don't know if it fits, the volcano plots or the QQ plots or the standard plots we have for you to choose on the resource, give us a call. I mean that. It's actually fun for us to build new portals. So if you go here to this tab at the bottom here, there's a demo on how to build your own portal. There's tutorials. There's a workflow. It's pretty straightforward. So I think that would be really great if you're interested in.